Okay, now first of all, we're going to look at ultrastructure. So what's mean ultrastructure? When you use the word ultra, means that not a basic structure only, that we can view it, for example, what we have learned in IGCSE or in SPM, nucleus, okay, cell membrane, cytoplasm. No, the one is the what we call this normal one. So, but now we're going to zoom it in to a very, very tiny detailed organelles of an eukaryotic cells. Okay, so what's it mean by eukaryotic cells? When you use the word eukaryotics, so if you break the word into two parts, the first part actually is eu, another one is karyot. Eu means true. Okay, in biology term, eu here means true. Karyot means nucleus. So it means that this kind of cells will have the nucleus. Okay, eukaryotic cell must have nucleus in their cellular structures. Okay, so but you have to be careful when you talk about this. When you talk about nucleus, some of the cells, they don't have nucleus. For example, red blood cell, no nucleus. Safety elements, they don't have nucleus. But those are matured red blood cells, those are matured safety elements. When they are young, immature stage, they do have nucleus. Are you clear? Okay, so means that in their lifespan, at any means they in the lifespan of the cells, okay, when they matured, so at some point in their life or span, they have nucleus. Okay, so when talk about eukaryotic cells, so they have a true membrane-bound nucleus and some other membrane-bound organelles. So very, very important why we need to have those membrane. Basically, we want to have the compartmentalization. Now, what's mean compartmentalization? You think about this. If there's no compartmentalization, there is a problem. You try to imagine, if you go to a house without compartment, you do not know where is a toilet, where is your living room, you do not know where is your bedroom, you do not know where is the kitchen. There is a problem. Why? Because now you want to sleep in your bedroom, but you don't know where to sleep. You want to go to toilet, you do not know where is the toilet because no compartment, correct or not? And you don't have a very conducive environment now. You try to imagine that in the room, you do not know where is the toilet, where is the bedroom. So it means that there is no conducive environment for you to rest in your bedroom. Study room, you don't have compartment. So always remember, I always use this example. Don't make your study room as your bedroom because the environment different. If you study in your bedroom, bedroom is for you to sleep. So it's the conducive environment for you to sleep. The function of the bedroom is for you to sleep and rest. So if you study inside your bedroom and you see the bed, definitely you feel like, oh, tired already. I want to sleep already. I want to rest already. Why? Because bedroom basically serve for you to rest. So your toilet, right? Can you see that you won't eat inside the toilet? Why? It's not conducive, right? Correct or not? It's not the appropriate place. So why our cells must have this membranous organelle? The idea here is to make it as many compartments as possible. What's mean as many compartments as possible? Means that at each compartment, we can have different kind of the activity and function. And within each compartment, you can have the activity, okay, simultaneously with other compartments. For example, you can rest in your bedroom. Someone can actually eat inside the dining room. Do together. We don't need to share. Can you see that? So each compartment will have their own environment. So nucleus will do its job. Mitochondria will do this job. Chloroplast will do its jobs. Are you clear? So means that they have their own conducive environment for the, or what we call it, ideal environment for the activity or biochemical activity to take place. Okay, so you can see that in this diagram now. Okay, you can see in this diagram, it's a typical animal cells. When I say typical means that it's not all animal cells, but generally all animal cells will have these structures. Okay, so what are the structure that we need to cover here is, first of all, the largest one and very prominent structure is known as the nucleus. Okay, we're going to start in study in our syllabus, nucleus. Second, we're going to follow by the nearest to the nucleus will be our rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay, then followed by what? Can you see that? Nearer to the rough ER, we'll have our Golgi apparatus. Then you can see that we have ribosome as well as the mitochondria. Okay. Then you can see that we do have the lysosome, centriole, we're going to learn. And then cytoskeleton, particularly we focus at the microtubules. And last but not least, our plasma membrane. Okay, now one structures that didn't mention here, one of the structure that is not mentioned here is the Golgi vesicle. Can you see that? The vesicle that formed from the Golgi apparatus is known as the Golgi vesicle. Okay, so how you differentiate between the structure, nucleus, a large prominent structure, and then if you look at this rough endoplasmic reticulum and the Golgi apparatus, you can see that they form something like a bag. Can you see that? Like a bag. So the difference between the rough ER and the Golgi apparatus, the rough ER, the RER, you can see that they are interconnected. Can you see that? They are interconnected. The bag, they are interconnected. Okay, the sac. Golgi apparatus, the bag, they are not interconnected. So this is how we differentiate them. Then you can see that rough. What's been rough? Because on the surface, can you see that? On the surface of the rough ER, we can get the ribosome. Can you see that? We can get ribosome, give them the rough appearance. How about smooth? Smooth ER, you won't see. Smooth ER, you won't see the ribosome. Can you see that? No ribosome there, right? So it means that this will be smooth ER. That's how we differentiate between them. Second, if a three-dimensional structure, you will realize that rough ER, they are bag. Can you see that? A sac, but smooth ER formed by the interconnected tubule. Can you see that? Interconnected tubule. Okay. Now, next, how we differentiate or identify the mitochondria? So mitochondria, you can see that inside you have convoluted, okay, something like a folded. Can you see that? The folding, okay, the folded membrane. So this is the structure of the mitochondria. Okay, so how you differentiate between the lysosome? Lysosome, basically, you can see that it's a round circular structure, okay, a spherical structure. And inside we have a lot of the enzyme. Centriole, you have one pair of them. Can you see that? Always two together, and they always perpendicular to each other. So one is horizontal, another one will be vertical. So this is how you differentiate, okay, the centriole. Microtubule, a little bit hard for us to identify them. You can see that they are something like proteins, okay, protein strands like this. Can you see that? Like thread-like structure, protein strands, so known as the microtubules. Okay, so inside the nucleus, okay, if we zoom in inside the nucleus, what you can see here will be a very prominent structure known as nucleolus. You want to label here, yes, okay, sorry. So this one, you want to label is known as nucleolus. So the membrane here is called nuclear envelope. Okay, it's a nuclear envelope. And then you can see that a lot of pore. So this one is known as a nuclear pore. And then you see the thread-like structure inside, right? Something like thread, right? So this one is known as the chromatid. Okay, known as chromatid. So what I label here, nuclear envelope, nuclear pore, nucleolus, and chromatins, all of them actually is termed as a subnucleolus. A subnuclear structure means that nucleus is the one, so subdivided into nuclear envelope, nuclear pore, nucleolus, and chromatids. Okay. So, Okay, so in our syllabus, as I said, that we need to interpret this ultrastructure through the drawing. So let us look at the answer now. So assume you guys have done this. So first, as I said, that this is a very prominent structure, the largest organelles. Can you see that? The largest organelle. So these organelles, basically, we know that is nucleus, okay? Singular called nucleus, plural called nuclides, okay? Nuclide, N-U-C-L-E-I, nuclide. So in this structure of nucleus, they have subnuclear, okay, subnuclear structures, which are, first one, can you see that? The prominent structure here, the very dark spot here, known as nucleolus, nucleolus, okay? So this is a spelling, nucleolus. So what is the function of nucleolus? Okay, generally the function of nucleolus is to synthesize, okay, or to make ribosome, okay? We're gonna learn it later, that's first. Second, you can see that, okay, this is the membrane, okay, something like membrane, something that actually surround, okay, something which is surrounding this, our nucleus. So this one is known as the nuclear envelopes, okay, nuclear envelope. And this nuclear envelope actually perforated with the pore, right? So this pore is known as a nuclear pore, okay, nuclear pore, okay. Then you can see a thread-like structure. Can you see a thread-like structure inside? So this thread-like structure is known as the chromatin. So later, I will explain more the difference between the chromosome and chromatins. Okay, it's like they are slightly different, chromatins and also chromosome. Okay, we will discuss this later. Okay, so now we have done for the first part, nucleus, a very prominent structure in the cells. Then you can see that, guys, near to the nucleus, we do have a structure. Can you see that? A structure, something like a membrane, they are interconnected. And you can see that the surface is a little bit rough. Can you see that? A little bit rough because of the appearance of the presence of the ribosome here. So we know that this structure is known as the rough endoplasmic reticulum, okay? Or in short, RER. So this is very important, guys. When you answer the question, if the question asks you to name the structure, you have to spell it full, right? You have to spell it full, rough endoplasmic reticulum. You cannot give me RER, okay? So now, we do know that endoplasmic reticulum, we have two forms. One is the rough one, another one is a smooth one. So smooth ER, actually, you can see that they are interconnected. Okay, this one is not sure, I mean, a little bit interconnected. So this one actually is termed as the smooth endoplasmic reticulum. Okay, smooth endoplasmic reticulum. Another one, you can see that. Can you see that? They're interconnected. So another one here is smooth endoplasmic reticulum. Okay. So now we do know that on the rough ER, we do have the ribosome. So ribosome have two forms. One form is attached. They are attached. Can you see that? Attached onto the rough endoplasmic reticulum. Another one, they are actually free moving, unbound ribosome. So we have two forms of ribosome. One is the ribosome on the RAR. Another one is the free ribosome on our, inside our cytoplasm. Okay. And then you can see that's something like a bag, right? Something like a bag, can you see that? And they are not interconnected. So the whole structure here is known as the Golgi apparatus, or Golgi complex. Okay. And because the Golgi apparatus always involved in the secretions, okay, by exocytosis, then you can see that this is a secretory vesicle or Golgi vesicle, right? Golgi vesicle or secretory vesicle. So sometimes it's a little bit hard to differentiate between the Golgi vesicle as well as the lysosome, okay, lysosome. Okay, a bit difficult to differentiate between both of them. Generally nearer, generally if nearer to the Golgi apparatus will be the Golgi vesicle. Okay, then you can see that something like infolded. Can you see that the folding of the membrane? So the folding of the membrane, this one we know that it must be mitochondrions, okay? Because one, mitochondria means plural, singular mitochondrions, plural mitochondria, okay? And then you can see that here a pair. Can you see that a pair? One is vertical, another one is horizontal, right? Vertical, horizontal, they arrange. So this one is centriole, okay? Centriole. And it points to the cytoplasm. So this one is a cytoplasm. And this one, the last one is the plasma membrane. So you can see that sometimes the plasma membrane form the folding like this, right? The folding. So this folding is known as the microvillus, okay? Singular microvillus, many of them called microvilli, okay? Microvilli. Do you manage to get all correct? Yeah, do you manage to get all correct? Yes or no? Okay, can you differentiate now? Do you need times? Okay, you need times to differentiate. Okay, so we're done for the animal parts, okay, the typical animal cells. Now, let us look at the plant cells, okay? So typical plant cells, you can see that on top of some of the structure, you can see that for plant cell, they will have chloroplasts, special for the plant cell, chloroplast. They have vacuole and the tonoplast, special, cell wall and plasmodesmata. Those are the structure. Okay, those are the structures special for the plant cells. So what are the structure that's special for the animal cells? Okay, so I'll come back to this. Lysosome as well as the centrosome, means centriole here, okay, centriole or lysosome. So these two, they are special for the animal. Means that you won't get lysosome and centrosome inside, okay, or centriole inside the plant cells, okay? So look at the structure. As I said that just now, prominent structure, nucleus. I mean, near to the nucleus with a rough ER, as well as the smooth endoplasmic reticulum. Then you have the ribosome attached to the surface. Okay, and then you can see that a convoluted one. Can you see that? Okay, the infolding of the membranes, which is a mitochondria. Okay, and then you have the Golgi apparatus and with the Golgi vesicle. Okay, then for the plant cells, we do have a special organelle here called chloroplast. So how to differentiate the chloroplast? You can see that they form a stack like this, okay? Something like stack. So this stack is known as granum or grana. Okay, something like you stack the money, right? There's the shilling one by one stack up. So this is how the structure look like in the chloroplast. Okay, so they have vacuole. So the vacuole is a large centrally permanently located at the center here. Is known as the vacuole. So this vacuole surrounded by the membrane. So very special here. Animal cells do have vacuole, but animal cell vacuole, the membrane is just called as a membrane. But for plants vacuole, the membrane is termed as the tonoplast. So it means that tonoplast, T-O-N-O, okay, tonoplast only appear in the plant cells. Animal cells membranes, a vacuole membrane, we don't give them a special name. Okay, so you have the cell wall. Then you can see the cell wall is perforated. Can you see the pore here? So this pore is known as the plasmodesma. Okay, so some structures are not labeled here. So let us label some of the structure here. So the pore is known as the plasmodesma. Okay, singular. Plasmodesmata, plural. Okay, more than one plasmodesmata. Okay, then the vacuole, the central located vacuole, the membrane here is known as tonoplast in this diagram. So highlight the structure that we need to know: nucleus, as usual, rough ER, smooth ER, Golgi apparatus, ribosome, mitochondria. Okay, and then in this case we have the chloroplast, which is surrounded by tonoplast. And then we do have the plasmodesmata on the cell wall, and you have the vacuole. Okay, cytoskeleton, we focus at the microtubule. Okay, then the last one, plasma membrane. So that's a good question. What's the difference between the plasma membrane and the cell surface membrane? Both of them are the same. Okay. So the cell membrane also termed as the plasma membrane or cell surface membranes. They are the same. Okay. So now, take 5 minutes, complete this diagram now. Okay, take 5 minutes, complete this diagram. So I assume that most of you guys have done the identifications. So let us look at the first structures. Now, in animal cells, nucleus will be the largest and very prominent structures. But in plant cells, not necessary, because the largest organelles that you can see in the plant will be centrally located to vacuole, and the vacuole will be very, very large. So this vacuole actually consists of two parts. The membrane, I said that the membranes of the vacuole for plant cells only, animal cells no. For plant cells only, the membrane is known as the tonoplast. Okay, sorry, the tonoplast, the membrane, and the structure inside, okay, is called cell septs. Okay, the fluids inside the vacuole is known as the cell sept. Okay, and then the second larger, very prominent structure here, you can see it, right? If you stain it properly, it will be nucleus, and we do know that for nucleus, okay, it has the subnuclear or subnuclear structures. So what are the subnuclear structures? As I said, that first you can see that this is a pore, so we have the nuclear pore. Second, you can see there a dark stain here, which is a very prominent structure inside the nucleus known as the nucleolus. Nucleolus. And then you can see the thread-like structure, right? So in the thread-like structure, so those thread-like structures is known as the chromatins. Okay, chromatins. And then this line point to the membranes of the nucleus, right? Membranes of the nucleus. So it's known as the nuclear envelope. So all of them belongs to the nucleus. Okay, now next, as I said that near to the nucleus you have the structure interconnected back with the dotted with a dot, right? So those dots actually refer to the ribosome. So means that this structure is known as rough endoplasmic reticulum. Okay, rough endoplasmic reticulum, and then the one that point to the dots. Okay, you can see that here we have also again point to the dot. So this dot is the ribosome that attached to the rough ER. This is the free ribosome. Okay, ribosome that attached to the rough ER, and we have the free ribosome. Okay, and then you can see that the structure here. Okay, we have the convoluted inner membranes. Okay, so we do know that this one must be mitochondrions. Okay, and then you can see that the stack of the back, a stack of the back like this, right? So this one is known as the Golgi apparatus or Golgi body. Okay, the smooth ER in this diagram not look at very smooth, but you can see that most of the part they don't have the dotted line, right? The dotted structure or the dots there. So therefore, this one is the smooth endoplasmic reticulum. They're interconnected, but it will compare to those of them. Can you see that? These two, they are rough ER. Okay. Now, what else we have? Okay, the green color of structure here. Okay, with something like with the lamella and the stack of the grana. So this one is called as chloroplast. Okay, then the rest of the structure, we do have this structure point to the cytoplasm. So this is a cytoplasm. And then where's the plasma membrane? Can you see that the first layer, this one? So this one is known as the plasma membrane. How about the cell wall? So this one is a cell wall. Now, how about this structure? The structure that glue, this is the first cell. This is another cells, right? So the one that glue both cells together is known as the middle lamella. Okay, middle lamella. Can you see that the center lines? that separate two cells. This is a cell wall. Can you see that? The one I highlight now, or point now, is the cell wall of this cell. Then you can see that the cell wall of another cells. So in middle of it, we call it as a middle lamella. Can you see that? The middle of it is known as middle lamella. So you can see that this is a gap, a pore at the cell wall. So those pore is known as a plasmodesmata, singular called plasmodesma. Okay, so I'll leave it a while so that you can check. Okay, so there is a question asked here. Okay, for the ribosome attached to the RAR, do we need to have write RAR in bracket next to it, or we just can write ribosome? Answer: you just need to write a ribosome, enough already. Full stop. Ribosome, full stop. Do not need to actually go and write ribosome, put a bracket RAR. No need. Okay, the structure is known as ribosome. Okay. So with this, I have done for the ultrastructures. Okay. So now we continue with the organelles. Okay, a little bit of organelles. What is the definitions of the organelles? So when we talk about organelles, organelle basically is a specialized subunit within a cell that has a specific functions. Okay, specialized subunits. So generally, they do have the lipid bilayer. Okay, so this lipid, if more precisely, is known as the phospholipid. Still remember what's a phospholipid? When we talk about phospholipid, basically means that lipid triglycerides, right? Normal triglycerides, but these triglycerides, okay, have the phosphate groups. Okay, so we're going to learn this one in chapter 2. Okay, the structure of the phospholipid. But if you use this as a definition, they must have the lipid bilayer. Some of the structure are not qualified as an organelle. For example, ribosome, right? But ribosome, they are not membrane-bound organelles. So, but they still carry out a specific functions. So therefore, nowadays, we separate them into two broad categories. So one broad category is called membrane-bound organelle. So membrane-bound organelles have two. One is a double membrane-bound organelle. Another group is called single membrane-bound organelles. Okay, so double versus single. And another group is called non-membrane-bound organelles. So example, you can write down. Okay, example for double membrane-bound organelles such as mitochondria, such as, again, nucleus, chloroplast. Single membrane-bound organelle, for example, RAR, SER, Golgi apparatus, lysosome. Okay. Non-membrane-bound organelles, ribosome, centriole. Okay, those are the structures. Okay, non-membrane-bound organelles and versus membrane-bound organelles. So those are the example. Okay. So again, now in terms of organelles, we differentiate them either membrane-bound or non-membrane-bound. If membrane-bound, so either double or single membrane. So if you double membrane, we have mitochondria, nucleus, as well as a chloroplast. Single membrane-bound, we have the endoplasmic reticulum, Golgi apparatus, lysosome. For non-membrane-bound organelles, ribosome, centriole. Another one you can put it here is vacuole as well. Okay, single membrane-bound organelles. Okay.